So I welcome you all in today's class. So I'm sorry this won't be an interactive class because I will not be able to hear you. So because we are joined through AgriDiksha, so this is the Government of India's initiative to so that everyone in any corner of the India or world can get these lectures. So today we'll be studying RFLP and AFLP in detail. So RFLP stands for Restriction Fragment Length Polymorphism. So restriction fragment length polymorphism is a marker that we use to see variation between two, say, crop plants or variants. So RFLP is a codominant marker. And before we move forward, this also gives us some bands, okay? And we should know how to interpret these bands. So if, for example, this is a gel, and this is, say, plant number 1, 2, 3, 4, 5, okay, and a band you see in each of these variants that you are assessing for a trait. So this band will be called as a monomorphic band. And the bands that are not found in all of them, okay, may be found in some, like this band is found in these two, this band is found in these two, but not in these. So these kind of bands are called as polymorphic. So the principle behind restriction fragment length polymorphism lies in the your restriction sequences or recognition sequences of a restriction enzyme. For example, I will say this is one DNA, okay, and this is another DNA. Say of two organisms or two different plants, and this is an example, okay? So for example, this is the total length of the genome, okay? Say this is 1000 base pairs long, and this one is also 1000 base pairs long. So for example, there is any restriction site. For example, a site is GAATTC, and this is the recognition sequence of EcoR1 restriction enzyme. And wherever this sequence will be present, and you give it with an enzyme or EcoR1, this will digest it, okay? This will digest it and cut this DNA that is having this recognition or a restriction site. However, if something happens that this sequence becomes GAAATC and GA, So what happened, you see this one is same, G is same, A is same, A is same, this T is same, this C is same. Only one nucleotide changed here. Now this EcoR1 will not be able to recognize this sequence and it will not cut this sequence because this is not the recognition sequence of EcoR1 because it has been mutated. Only one mutation has abolished a registration site. Or other thing can happen, an inverse can happen. For example, in this DNA you have a site GAA TTG, for example. And you know that this is the complementary strand, and you know that this is not the recognition sequence of your registration enzyme, and EcoR1 will not cut here. But if what happened that there was a mutation and now this recognition sequence is not a recognition sequence of your registration enzyme. So if you cut this DNA with a EcoR1, it will not cut because this is a sequence that does not, which is not recognized by your EcoR1. But however, if again a mutation happened, and mutation happened in such a way that this G became C, so this became a C, now this will be recognized by your EcoR1 restriction enzyme. That means here what has happened that a restriction sequence has been added, which was not there before. So one mutation at one place. Now you have two cases, if we can. So now you know that so either a restriction site can be abolished or a restriction site can be added. Both things can happen. Now we take the both these cases again. So you have organism number 1 and organism number 2, and say the length is 1000 base pairs. And this is also 1000 base pairs. And what has happened between these two organisms, say one restriction site is here, okay, and one is here. Say for example the fragment size is 1000 base pairs, and from here to here you have say this is 600 base pairs and this is 100 base pairs. So total sum will be your 6 plus 3 is 9 plus 1 is 1000 base pairs. In this case what has happened, so this for example say is EcoR1. We are treated with EcoR1, and this EcoR1 has two sites recognition sequence in this organism, and here also it's the same. However, one of the sequences has been, one of the recognition sequence has got mutated, and because of mutation now it's not recognizable by the or recognized by your restriction enzyme. So for example this sequence got abolished and it cuts only here, okay. So this is 600 base pair, and this fragment is your 300 plus 100 is 400. Total is 1000. So what is the difference between these two? The difference between these two is that here you have a restriction site that got abolished, and now this restriction site is not here. So this is difference between two organisms. So it mutated. So mutations can be, for example, you have say this is a purine, A and G, and this is a pyrimidine, C and T. So if a purine changes into a purine, A mutates into a G, or a G mutates into an A, or a pyrimidine changes into a pyrimidine, say T mutated to C or C mutated to T. These types of mutations are called as transitions. So since you have 4 nucleotides, you can only have 4 transitions. C to T or T to C, A to G or G to C. So these are 4 transitions can happen. So this mutation can have occurred because of this. Or a purine changes to a pyrimidine. Say A changes to C or C changes to A. A changes to T or T changes to A. T mutates to G or G mutates to T, or C mutates to G or G mutates to C. So this, when a purine changes to a pyrimidine or a pyrimidine changes to a purine, this is called as transversion. These are the main 2 types of mutations, and you can have 1, 2, 3, 4, 5, 6, and 7, 8. You can have 8 transversions and 4 transitions. So mutation can occur because of these. So we'll not go to that detail, that's not our topic, but so now the mutation occurred. Now if we run them in a gel, we have restriction digested, and now just run the DNA in an agro gel electrophoresis, and what will happen? So this is your gel, this is your marker or ladder, this is your number 1, and this your number 2 lane. Okay, these are lanes. So this is one DNA we load here, second DNA we load here, and this is a marker that will tell us 2, 3, 4, 5, 6, 7, 8, 9, 10. That will tell us this is 1000 or 1 kb, and this is 0.1 kb or 100 base pair. 100 base pair. This is 200 base pair. This is 300 base pair, 400, 500, 6, 7, 8, 9, and 10. So if we run this DNA that we have cut with the EcoR1 on this gel, so what we'll get? We'll get a band at 100, at 1 we'll get at 300, and other we'll get at 600. So this is 600, so 1 band we got here. This is 300, 1 band we got here, and 100, 1 band we got here. Right? Now, in this case, since one of the restriction sequences was abolished, it cut only at one place, okay? So the bands, it made a 400 base pair and a 600 base pair. So you have a 600 base pair that will be here, and a 400 base pair that will be here. Now you can see the difference in the binding pattern between these 2 organisms, and what gave rise to this difference in the binding pattern? A mutation at the restriction site. So restriction, fragment, and polymorphism can tell us the difference between 2 organisms based on the either incorporation of a restriction site or abolition of a restriction site got abolished. However, so this is the concept. In reality, this does not happen. Why this does not happen? Because if you take the genomic DNA from any organism and run it on a gel, it will give one band next to the well, because genomic DNA is very heavy. It will not move further in the gel. Okay, it will be stuck at near the well. However, when you restriction digest it with EcoR1, any DNA, you will get a smear. Smear means you will get fragments of each sizes. In this case, case 1 and case 2, again when you did this, you will again get a smear. So by restriction digesting the 2 DNAs, when you run them on gel, you do not see the difference. This, what I told you, was the concept that what happens actually. But this you will not be able to detect on a gel. So now how you can detect it on a gel? So what we do once we run this gel, we use a technique we call as southern blotting. We use southern blotting, and southern blotting uses a probe. A probe is a short nucleotide that is complementary to any DNA region, okay, any DNA sequence, and it binds to that sequence. So, for example, we use a probe here for say 100 that binds to a 100 base pair, and that's say fluorescent. That's a fluorescent. We tag it with a fluorescent, and it's a DNA molecule. It will bind to a region in the 100 base pair, because we can design probes for a particular region. Here we have designed it for this 100 base pair. जहाँ पर 100 base pair होगा, वहाँ पर ये चिपक जाएगा और एनील हो जाएगा, एंड वहाँ पर फ्लोरेसेंस होगी तो हमें दिखेगा. तो साउदर्न ब्लॉटिंग में हम क्या करते हैं? We take this gel, okay, and put a nitrocellulose membrane on it. Or a nylon membrane also. There are many membranes. So this is nothing but, say for example, you have a gel, and this nitrocellulose membrane is just like a sheet of paper. So you take it and put it on the gel once you have run the gel. So this nitrocellulose membrane is positively charged and your DNA is negatively charged. And so what we do, say for example, इस gel को हमने ऐसे देखा, ठीक है, from the side. So this is your gel. इस पर हमें अपना nitrocellulose membrane रखा, ओके, एंड उस पे हमने बहुत सारे ब्लॉटिंग पेपर्स रखे, और हल्का सा वेट इस पर रख दिया हमने. And we can keep it overnight, or आजकल ट्रांसफर मशीन है जो जल्दी ट्रांसफर कर देते हैं. What we want to do here, we want जो gel DNA में जो हमारा DNA है, that will get transferred to the nitrocellulose. So due to a process called as capillary action, since this is a gel, there are blotting papers, so the water will try to move upwards through capillary action, और इस केस में क्या हो जाएगा, जो DNA आपका है वो भी आपका nitrocellulose membrane पर चिपक जाएगा. So now you have a situation So now you have a nitrocellulose membrane. You have a nitrocellulose membrane where you can see nothing. Okay, the DNA is there, but you will not be able to see it. So what we do now, there's a process for southern blotting. So we use buffers, we denature the DNA because the DNA is double-stranded. Okay, we can also cut the DNA or pulverize it using different buffers. So that different DNA breaks into different smaller fragments, and it's easier for your probe to like it. Then we mix it with your probe. Probe is nothing, it's also DNA with a fluorescent tag or a substrate. So now what will happen? So this 100 base pair will be somewhere here in the gel, right? Okay. In this case, this 100 base pair will be here. But in this case, this 100 base pair is a part of this 400. You see 300 plus 100. So the gel did not cut this 100 base pair, जहाँ पर हमने probe बनाया है, that will be a part of this 400. That means वो आपका यहाँ पर होगा. वो आपका यहाँ पर होगा. So once you add the probe will bind to your DNA. इस केस में यहाँ पर bind होगा. में यहाँ पर bind होगा, और probe इस केस में यहाँ पर bind होगा. Now since probe has a fluorescent label with it, जब हम इस पर एक्सरे फिल्म रखेंगे, ओके, या फोटोग्राफिक फिल्म वगैरह, तो क्या होगा, ये एक्सपोज हो जाएगी फ्लोरेसेंट डाई के, और वहां पर एक्सरे फिल्म पर हमें फ्लोरेसेंस मिलेगी, जो फ्लोरेसेंस इसको फोटोग्राफिक प्लेट पर अपना ये छोड़ेगी, क्या बोलते हैं, इंप्रेशन। और जब हम इसको देखेंगे एक्सरे फिल्म को, हमें दो बैंड्स मिलेंगे, एक यहां पर मिलेगा, एक यहां पर मिलेगा। नाउ अब देखो, अब जो बाकी सारे बैंड्स हमें नहीं दिखेंगे, हमें वही सब दिखेंगे जहां पर हमारा प्रोब बाइंड होगा। और प्रोब हमने कहां पर किसके लिए बनाया था, इस 100 बेस पेयर के लिए। सो प्रोब किसी भी जीन के लिए बना सकते हैं, किसी भी तरीके से बना सकते हैं, दैट डिपेंड्स ऑन यू। बट हियर आई एम टेलिंग यू दैट नाउ यू आर एबल टू डिफरेंशिएट बिटवीन two organisms vis-a-vis your restriction fragment length polymorphism.